Hey, Hammy here. Coming today with the first part of section 8.3, in which we will begin to look at some practical applications of biotechnology. In other words, using technology to edit and change genes. So the first part here, we want to look at some of the methods that scientists use to work with and manipulate DNA. Okay, kind of the technical definition of biotechnology. Remember that bio means life, and so we're used to talking about technology. Okay, those technology are things that make our life as humans easier. So we program them to do things or make things easier. So when you add the prefix bio to the front of it, it's going to have to do with kind of life and living things. And a lot of times it refers to working with the genetic makeup. Two big areas we'll look at in later videos are agriculture, okay, in order to create crops. Okay, a lot of our seed bags we use on our farm, something like this. Notice down here, technology that yields, okay, from Pioneer. And if you look over here, you can see some of the Liberty Link, Hercules, some of those traits that they've put into these corn plants, into the genetics, to make it easier for the farmer to grow them. And the second one we look at is medicine, whether that's gene therapy and also stuff for human diseases, and how can we prevent them and how can we treat them. One of the big things that people think about is cloning. Okay, now cloning, like reproductive cloning, is cloning a whole organism, and we'll take a look at that in a later video. On this step, one of the first methods that scientists do is gene cloning. In other words, cloning or making copies. So finding that one gene that we want to work with, how do we cut it out and then isolate it, make copies of it, and put it in somewhere else? Okay, well, it usually takes about 4 steps. The first one is isolation. We find that gene where it's at, and we need to isolate it. We need to remove it from the rest of the genome that we're working with, whether that's human or an animal or a plant. In order to do that, there's these little DNA scissors called restriction enzymes, sometimes called restriction endonucleases. Most of the time you'll hear it as restriction enzyme. Restriction enzymes usually target a specific base sequence or a recognition sequence. Okay, recognition sequence. And it's always the same letters, and it tends to be a palindrome, or we say palindromic. Okay, remember, a palindrome is like race car. It's the same one way as the other. And a lot of them make kind of these sideways cuts. Okay, so like this would be, I think, ECO-R1. Okay, ECO-R1. The restriction enzymes are often named after where they're found. Okay, they're used in bacteria. They first found them because bacteria use them. So when a bacteriophage inserts its DNA, these little restriction enzymes would cut up these enzymes and destroy, or cut up the DNA and destroy it before the viral DNA could take over the bacterium. Okay, so, but we use them because we learned that they have specific sequences that they cut the DNA. So ECO-R1 is going to cut between the gene A, go across the A's and T's, and then cut down through here. Okay, so it's named after E. coli strain R1, the first enzyme we isolated from them. So it's kind of named after that. HIN3 is another enzyme, and there's many of these. And you can see kind of how it would cut through that double helical DNA. Okay, and this cut is important because it leaves exposed bases here. Those exposed bases are important for step 2, that's ligation, okay, or using DNA ligase to stick them back together. Remember, DNA ligase is like the DNA glue. Okay, so notice here, sticky ends. What we do is we cut the piece of DNA we want, like say it's a human gene for insulin. Okay, we'll cut it out of the human with the same restriction enzyme as we cut it out of a plasmid. Plasmid are these little circular rings of DNA often found in bacterium, kind of not their big loop of DNA, but they have these little tiny rings as well. a little, we call it accessory DNA called plasmids. We will cut that with the same restriction enzyme so that the sticky ends will match up, okay? And then when we mix these two together, okay, the sticky ends will match. Okay, that human insulin gene will now be inserted into our plasmids, and we'll use DNA ligase to kind of seal up those genes within the plasmid. Step 3, okay, step 3 is transformation, getting that recombinant DNA, often written rDNA, okay, or sometimes you'll see it REC DNA. Okay, stands for recombinant DNA because it's recombinant. It's DNA from two different sources put together. So again, down here they're showing growth, human growth hormone. Okay, cut out of a human cell's genome. Okay, we take a plasmid out of the bacteria, cut it with the same enzyme, EcoR1. Okay, notice the sticky ends will match. Get DNA ligase to put them together. Now we will use this recombinant DNA or recombinant plasmid, put it back into the E. coli, okay? Little E. coli bacteria here, get it back into the bacteria so it will start reading that gene and start producing human growth hormone. Okay, so remember transformation, and when we change an organism this way with foreign DNA, okay, we have a bacterial cell with human gene in, okay, that's where we get this name genetic engineering. Okay, we are engineering the DNA. We're building and putting pieces together. Okay, step 4, final step is selection. Okay, now we have this DNA, these plasmids, the transformed plasmids and bacteria, we need to grow them. Okay, but usually when you're mixing the recombinant DNA and you're cutting with sticky ends, do the plasmids ever just go back closed? Yeah, sometimes. Do all of the bacteria pick up the new plasmids? Not all of them. So we need some way to select for just the ones that have our recombinant DNA. A lot of times when we do this in lab, in BioAP, we transform bacteria with a gene for a green fluorescing protein, GFP, and it's on a P-glow plasmid. Okay, and when shine it under UV light, it shines this bright green color. Pretty cool. Okay, well, along with that, on that plasmid, we also put an antibiotic resistance gene. In our case, we use it for ampicillin. Okay, so when you put that into the bacteria, so normally ampicillin would kill the bacteria. When we put in our recombinant DNA plasmid, it has a gene for ampicillin resistance. So now when we transform our bacteria and grow it on a Petri plate with ampicillin, okay, the recombinant DNA, the transformed bacteria, are antibiotic resistant and will grow. Guess what will happen to all the others? They'll be killed by the ampicillin. So we are selecting for just the bacteria that we changed, and they are the only ones that will grow on this plate. So we know when we can prove it by shining a UV light on it, that they are the only ones expressing those genes. Okay, they are the only ones that growing are the ones that have taken up our recombinant DNA. Another step scientists use is polymerase chain reaction, often known as PCR. Okay, what we do with PCR is we use it to kind of like a photocopy machine of a gene. So once we cut out a gene that they want to work with in a lab for experiments, they'll use PCR to make many copies of this gene in a short amount of time. Okay, how that's done usually in 3 steps. Step 1, they need to denature the DNA. Okay, the DNA is heated. Okay, by heating it, remember that there's hydrogen bonds between the A's, T's, G's, and C's down the middle of the molecule. That heating will break those hydrogen bonds, which will separate it. So here at 95 degrees, almost to boiling. Okay, remember 100 degrees Celsius is boiling. Okay, and then that separates these 2 strands of DNA. Okay, and then the second step is annealing. Annealing is they're cooled back down to 55 degrees, and then we add DNA primers to both ends. Okay, add DNA primers to both ends so that we can begin replicating or adding new base pairs there. But remember from the DNA replication video, and when we studied that in that chapter, that it has to have that primer before it can begin building. Okay, and this third step, okay, is once we have the primer laid down, the third step is extension, okay, where it's warmed back up again, and we use what's called Taq DNA polymerase. It's polymerase we actually found in the Yellowstone hot springs, these guys right here, which is Thermus aquaticus. Okay, that's what Taq stands for, Thermus aquaticus, which is a bacterium, these little guys here that we found in the hot springs that work well at hot, very warm temperatures, and they just start building. Okay, they start adding new nucleotides onto there, and we make 2 new strands. Okay, and it just kind of keeps going, keeps going, keeps going. And what we do is these 3 steps, okay, we repeat, not reread, repeat. Okay, we repeat steps 1 through 3 over and over again. So then the next cycle, okay, so we went from 1 strand. After 1 cycle, we have 2 strands. After 2 cycles, 4 strands. Okay, after another cycle, 8 strands. So it starts to multiply this gene that we want to make a copy of it. It multiplies it exponentially. And they'll put these in a thermocycler PCR machine that will go run overnight. And after, in the morning, they'll have millions of copies of their gene that they can work with in their lab. And it's actually quite easy. And we have very expensive machines that will do this for us nowadays. A third tool that scientists use is called gel electrophoresis. And what we do with electrophoresis is we chop up our DNA into pieces, and then it separates the mixture based on size. Okay, this little pieces will wiggle faster than the big pieces. So what we do is we take our sample, okay, and we cut them usually with our restriction enzyme. Okay, so we cut it into pieces, and then we load it into the gel, into this end here that has these little pits and these wells. Okay, the gel is going to be almost like jello-like substance with little holes cast in it. When you pour the gel, you have these little comb that sticks in there. When you pull it out, it leaves these little empty wells. You put your DNA samples down in there. Okay, you attach it to a power source, and you run electrical current through it. Okay, DNA is negatively charged, so you put the negative electrode over here, and you run electricity through that gel. Okay, and by doing that, the DNA is going to try to wiggle towards the positive end, and the little pieces can wiggle through the gelatin much faster than the big pieces, so it separates them out based on size. Okay, so here you kind of see a setup over here where you have your wells right here and a little power supply over here, and it's running electricity through this gel like this. And when you get done, okay, when you get done with your gel, we often will use some kind of stain that attaches to the DNA, and we stain it, and we can take pictures of it, and now we have a picture of all those little pieces of DNA. What do we use gel electrophoresis for? Well, one big thing is in forensics. Okay, what we would say forensic fingerprinting. Okay, where we find DNA at a crime scene. Okay, and then we have several suspects. Okay, we compare the crime scene because some of it might be the victim's DNA, and then we look at those banding patterns. So look at the crime scene here. Then we look at the banding patterns of our possible suspect 1 and suspect 2. Which suspect do you think did it? Well, look at the banding patterns here, 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 here. Look at there, it matches up. Darker line here, darker here, and on and on and on. Okay, so based on the DNA, we would pick suspect 1. We can also use it for paternity tests. Okay, where we have a mother and child, and we want to see which the father is. Okay, now it's a little bit more confusing because the offspring will have some of the mother's DNA and some of the father's DNA. Okay, so you look at the offspring. Look, the mother does not have that band here. So we look across here. Look over here. Okay, male 2 has that band. Okay, the next one for the offspring matches up with mother. The next one does not match up with mother, but look over here, male 2. The next one, male 2. Okay, these two match up with mom. Okay, this one matches up with mom. If you look at this one, come over here to male 2. So if we were to, make a conclusion, we would say probably male 2 was the father. The other thing we can do is to isolate our genes. Okay, so scientists will take certain sections of DNA or genomes and cut them up and run them through the gel electrophoresis. And then they have a process called southern blotting technique. That gets a little complicated, but they just take this gel and they attach it to a film, okay, and they can draw the DNA up out of the gel. And then let's say they want this gene right here. Okay, they can just take a little knife and they can cut out that DNA, and they have isolated that gene, okay, from all the others in the genome. And then they can get it out, and then they can run it through PCR, which we just talked about, and make a lot of copies of it and use it for their experiments. Okay, so these are just some practical examples of what we would use gel electrophoresis for. One of the last really cool tools is what we call CRISPR, or sometimes you'll see it called CRISPR-Cas9 is kind of the one you'll hear a lot about. This is a little enzyme that we found again in naturally occurring, okay, in bacteria, and it was a defense system they used for bacteriophages that were trying to insert their DNA. And this thing would go in and it would cut them up, and it would also cut at specific sequences so it didn't cut the bacteria's own DNA. Well, we've taken these little enzymes and we've reprogrammed them to target specific stretches of DNA. So if we sequence the gene and we know what it is, okay, we've taken this enzyme, this kind of an animation of it down here. Okay, it will find that stretch on the DNA and it will go in there and make a cut. And we've even modified it and we've added other enzymes to it, so it might add a gene in, or it might cut it out completely, or it might go in and just change a couple base pairs, okay, and alter it. Some big implications when it comes to things like cloning and gene therapy and stuff like that, and many treatments like for cancer and other genetic disorders. You'll see kind of an infographic on the back of your note packets, and we'll look a little bit more at this CRISPR and its possibilities in our class discussion as well. So I hope you kind of looked or learned some of the new tools that scientists are using in order to manipulate and work with our genome and the chromosomes and the DNA, and to get out the genes they want and how they can find them and put them in other things and kind of work with them.